There are 4 phases in the cell cycle. The G1 phase is the growth phase. Epidermal growth factor initiates DNA transduction and a response by binding with the RTK. The cell responds to the binding by starting the S phase. DNA is synthesized and replicated in this phase. The G2 phase is another growth phase, but the cell prepares to enter mitosis. And finally, the cell division occurs in the M phase. During the M phase, the cell is visibly dividing and undergoes mitosis and cytokinesis. The cell that is divided into 2 cells in the M phase will again enter the G1 phase and repeat the cycle.
Above are the bases, enzymes, and other elements of DNA replication. DNA is made of 2 anti-parallel strands.
DNA is made of 2 intertwined strands of nucleotides connected by covalent sugar phosphate bonds between one phosphate group and another's deoxyribose. Each nucleotide is made of a deoxyribose with 5 carbons that count clockwise from the O. From there, it goes carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. It is attached to a phosphate group in a nitrogenous base. In DNA replication, these bases include adenine, thymine, guanine, and cytosine.
DNA replication starts at the origin of replication. The helicase enzyme then unzips the DNA strands by breaking the hydrogen bonds between the bases. Thus, it helps in the formation of the replication fork. And at the origin of replication, the enzyme primase comes to help the synthesis of the RNA primer, complementary to the DNA template strand. They catalyze the synthesis of short RNA molecules used as primers for DNA polymerases. Next comes DNA polymerase III, which is the main enzyme which catalyzes the DNA synthesis, while DNA polymerase I and II are involved in DNA repairing and proofreading. DNA polymerase adds nucleotides complementary to the template DNA. However, it is facilitated by the small DNA or RNA fragments of primer. The other new strand, which runs 5 to 3 away from the fork, is trickier and it is made in fragments. This strand is called the lagging strand and it is made of Okazaki fragments. However, the gaps between the DNA fragments are sealed by DNA ligase. You now have 2 strands of DNA so the cell can replicate. This ensures that each daughter cell gets a copy of the genome and therefore successful inheritance of genetic traits.